Welcome back, everyone, and this is the beginning of our discussion on energy and biological systems and our section on metabolism. So in this particular presentation, we're going to talk about concepts related to energy and biological systems. We'll talk a little bit about chemical reactions and the types of chemical reactions you'll see, and then we'll start our discussion on metabolism. We're going to focus on glucose catabolism, or the breakdown of glucose, or cellular respiration, as you may have learned about in the past. So energy transfer occurs starting in the environment. All energy really comes from the sun. It comes from radiant energy, heat energy. Plants use this energy to trap the energy in biomolecules, and that energy stored in biomolecules is ingested by animals and humans, and that energy is in the form of glucose, and we can use that energy for work and for life. So it's mostly that the sun has this energy. It ends up getting trapped in biomolecules, and that energy is actually stored in the chemical bonds of those biomolecules. And then animals eat the plants and use that energy, or sometimes we store it if we have an excess of energy. When we talk about energy, energy is the capacity to do work. Living organisms need energy to complete various types of work. That could be in the form of chemical work, so all the chemical processes that happen within a living organism. Transport work, so the ability to move a molecule from one part of the body to another part of the body, as well as we need the energy to do mechanical work, so muscles for energy for movement, energy to contract muscles, and we'll get into that later on in this course. So chemical work, making and breaking chemical bonds. It really enables the organisms and the cells within the organisms to grow and maintain that stable state within the body, or homeostasis. The transport work is in the realm of ionic movement and molecular movement, so moving sodium, moving potassium, moving glucose from one compartment of the body to another compartment. This type of transport work is very useful in creating concentration gradients. And we're going to talk very heavily of concentration gradients in a couple of sections of this course, mainly the renal section, as well as sort of indirectly in the respiratory section. Mechanical work, it can be at the macroscopic level, like I mentioned, so the contraction of muscles. But it can also be at the microscopic level, so movement of organelles within cells requires energy to be put in to actually do that mechanical work. So there are a couple of types of energy that you have probably heard about before. So there's potential energy. That's the energy that is the type that is stored. So if you think of a ball at the top of a ramp, that ball has stored energy, and that stored energy can be converted to energy of motion, or kinetic energy, and that's the second type of energy. When the ball is rolling down the ramp, that is the energy of motion. The first image is actually showing somebody pushing the ball up the ramp, and that is called work. So we're pushing the ball up the ramp, and so actually a force multiplied by a distance and pushing it up the ramp allows that ball to store energy, either in the form of potential energy if it's staying at the top of the ramp, or that kinetic energy if it's rolling down the ramp. Energy transfer in biological systems is not 100%, and in almost all circumstances, whether it's biological or not, energy transfer is not 100%. In biological systems specifically, some of that energy is lost in the form of heat, and this occurs in the human body as well. The majority of the energy used in physical activity is actually lost as heat rather than transformed into work for muscle contraction. And it's about 70% of that energy is actually lost in the form of heat. Even in non-biological systems, if you have energy of, say, billiard balls, when a billiard ball hits another billiard ball, there's a sound that's created. Some of that energy of the moving billiard ball is converted to sound energy. So not all of the energy will be converted to or transferred to that billiard ball that it hit. And so, but when we think about it in biological systems, the majority is lost as heat rather than actually being transformed into the work component. A few laws of thermodynamics to sort of hang our hat on for when we talk about energy. First law of thermodynamics is that the total amount of energy within the entire universe is constant. So energy cannot be created, energy cannot be destroyed. Therefore, the human body can't create energy. It must be imported from the outside, and that's in the form of nutrition. So we get our energy sources from the external environment. We can't create energy out of nothing. Second law of thermodynamics is really just a process whereby the universe tends to move from a state of order to disorder. And this is sometimes referred to as entropy. And if you think about a living organism, we'll generally maintain a sense of order within the living organism. In order to maintain a state of order and to overcome that entropy or that eventual disorder that naturally happens, a living organism requires a constant input of energy to maintain that state of order. Few examples of disease processes that occur because of an inability to maintain a state of order for the entire lifetime would be something like arthritis. Our joints tend to go from a state of order or working very well, having that synovial fluid between them, and having that smooth surface for joints to very easily glide over each other, for movement to be very seamless, to a state that later in life, you end up getting breakdown of those joints, and the joint space begins to get narrowed, and rather than a lot of synovial fluid separating the two bones that are involved in the joint, those two bones start to make contact with each other. And that contact that they make causes there to be some degeneration of the joint space and will lead to pain. And that's an example of going from a state of order to one of disorder or entropy. So chemical reactions in living organisms do one of two things. It either transfers energy from one molecule to another molecule, or it uses the potential energy that is stored in a molecule. And so an example of that would be larger molecules have more chemical bonds, and therefore when you break those larger molecules down, you're going to yield more energy. So a large glycogen molecule, glycogen as we know is that polymer of glucose in animal cells, that polymer of glucose or glycogen, if you were to break down that entire molecule that entire molecule of glycogen, you're going to get a lot more energy than if you just break down a single glucose molecule. The more chemical bonds you have to break down, the more energy that you can release. So chemical reactions, whether we're talking about biological systems or not, chemical reactions need a certain amount of energy in order to get started. And that type of energy, that energy is called activation energy. And it's an input, an initial amount of energy that's required to start off a reaction. It sort of pushes the reaction to start. So if we imagine this chemical reaction where this person is pushing the ball up a ramp to the top, and then that ball can then freely fall to the bottom of the ramp. That ball must be pushed up the hill before it could roll down the slope by itself. And that, if you think about pushing the ball up the hill, that would be the energy that's required to be put into a system, or the activation energy, before the reaction can even happen. A couple of types of chemical reactions. There is a type of chemical reaction called an exergonic reaction, whereby the amount of energy in the reactants is a lot higher than the amount of energy in the products. And what that signifies is that there's a certain amount of energy, if this reaction were to happen, a certain amount of energy that would actually be released and would be available to do work. So, an example of this type of reaction would be an ATP molecule reacting with a water molecule, and the hydrolysis of ATP will yield an ADP molecule plus an inorganic phosphate, and this amount of energy that's released. So when that energy gets released, we know therefore that the products are going to have less energy than the reactants did. And this is an example of an exergonic reaction. There's the opposite type of reaction called an endergonic reaction that really traps energy, because the products have a higher energy than the reactants do. And so an example of this type of reaction would be the synthesis of glycogen from many glucose molecules. Glycogen has more energy than the original glucose molecules did, and therefore the product has a higher energy than the reactants. So exergonic products have a lower energy than the reactants. Endergonic products have a higher energy than the reactants. So therefore, you know that you have to input more energy into the system in order to build those products in an endergonic reaction. We tend to couple these reactions in biological systems. So the energy that's released from an exergonic reaction, like the breakdown of ATP, can be used to fuel an endergonic reaction. And that happens very often in biological systems. We couple endergonic and exergonic reactions. And so in this case, you have the endergonic reaction happening between E and F, making the higher energy products G and H, and the energy that was input to the system to allow for that reaction to occur came from the breakdown of ATP, which is that exergonic reaction. So cells can also trap energy temporarily in the form of high energy electrons. So while they're waiting to be put into chemical bonds, this energy can be temporarily stored in these high energy electrons. And you've probably heard of some of these molecules before. So you've probably heard of NADH. That's a molecule that stores high energy electrons. There's FADH2. Both of these molecules are involved in cellular respiration, or the glucose catabolism or breakdown of glucose. And there's another molecule called NADPH, also involved not in cellular respiration, but in a lot of biochemical reactions within the body. We're going to focus on the first two when we discuss cellular respiration. So energy that's released by the exergonic reactions is generally trapped in a high energy molecule. And so when you have an exergonic reaction, the energy released is either going to fuel an endergonic reaction, which we've talked about, or some of it's going to be released as heat energy, so it's going to be lost. We can't actually do anything about that. Or some of that energy that's released is going to be in the form of these high energy electrons. And these high energy electrons actually have to be stored in specific molecules, which we just talked about. And we're going to focus on NADH and FADH2. That NADH and FADH2, then the high energy electrons are used to produce ATP. And that ATP energy can be utilized to allow for movement. It could be used for endergonic reactions, whereby you need an input of energy for the because the products have a higher energy state than the reactants do. But a lot of the time this energy that's released is stored in these high energy electron molecules. Most biochemical reactions in the body won't occur if they were just left to happen on their own. It's very unlikely that they would occur. So most reactions in the body require an enzyme in order to increase the rate of a reaction. Enzymes normally will just bring substrates into the best position in order to react with each other. And by bringing them in the best position for the reactants to, the substrates to react with each other, it very significantly lowers the activation energy. So the energy that you actually have to put in is a lot lower than if you did not have the enzyme at all. Most enzymes, when you hear their name, and I think I mentioned this already, most enzymes have the suffix ASE, ACE. And so if you ever see that at the end of a molecule, it's mostly referring to an enzyme. There's another piece of nomenclature that you should be aware of. If you see the ending kinase, it almost always means that there has been a phosphate group added to one of the substrates. So if you see an enzyme named kinase, always ask yourself, where's the phosphate group been added? And we'll see examples of this a little bit later on today. So a lot of different types of enzymes that exist. There are or enzyme reactions that exist. There are redox reactions or oxidation reduction reactions. These types of reactions in the body are most important in energy release and transfer. So if you remember from your chemistry days, you remember the phrase Leo says Ger, loss of electrons is oxidation, gain of electrons is reduction. That's what's occurring in these types of reactions. And while the electron is being transferred from molecule to molecule, some of that energy in the electron is going to be transferred. There are hydrolysis and dehydration reactions. We've seen examples of these already. These are most important in breakdown reactions and synthesis reactions of large biomolecules. So when we make sucrose from glucose plus fructose, this is a dehydration reaction. The reaction between glucose and fructose will cause a water to be released, a water molecule, and therefore it's a dehydration reaction because water's lost. It's dehydrated. There's addition, subtraction, exchange reactions whereby you either add or remove or exchange a specific piece of a biomolecule. An example of an enzyme that does this is a kinase enzyme that will add a phosphate group to molecules. If it goes in the reverse direction, it removes a phosphate group. There are also ligation reactions that will join two biomolecules together. So if you remember, if you've learned the citric acid cycle, there's this molecule called acetyl-CoA. There is an enzyme that will ligate the coenzyme A with the acetyl group. And that is a ligation reaction. So we'll move into metabolism now. Metabolism just stands for all the chemical reactions that take place in an organism. And this can be separated into two specific arms. There are the anabolic reactions or anabolism, and this is these types of reactions result in the synthesis of larger molecules. Catabolism, on the other hand, is the other arm of metabolism, whereby the energy will be released from the breakdown of large biomolecules. So I remember these by thinking anabolism. I think of anabolic steroids. person builds up muscle. And catabolism, I think of catastrophe. Everything just sort of breaks down. So that's how I remember the two arms of metabolism: anabolism and catabolism. Catabolic energy, energy is either released or stored. And so this energy is either released or stored in one of two places. It's either released or stored in the bonds of ATP, so those high energy phosphate to phosphate bonds, or it is stored in the form of those high energy electron molecules, so NADH, FADH2, NADPH. Anabolic energy, on the other hand, energy from those molecules like ATP, FADH2, and NADH is stored in the covalent bonds of biomolecules. So when these substances, when ATP gets hydrolyzed, that energy is used to catalyze a reaction to build molecules up. And that energy from the release of the phosphate to phosphate bond, from the breaking of that phosphate to phosphate bond in ATP, will end up getting stored in these large biomolecule chemical bonds. And so that's anabolic energy. Metabolic pathways in general, whether they're anabolic or catabolic, tend to proceed in sequence. Every step of the pathway proceeds in a specific order. And each step in the pathway, this would be the reaction going from A to the end product D. Every single reaction that's happening will be catalyzed by a different enzyme. A different enzyme is going to allow the conversion from A to B. Another enzyme is going to allow the conversion from B to C. And a third enzyme will be allowing for the conversion from C to D. The molecules, while we're just going from A to D, the molecules in between, that B and C, are referred to as intermediates. And some of these intermediates, they are just byproducts of the metabolic pathway. But some of these intermediates can also participate in other metabolic pathways. If they can participate in other metabolic pathways, we call those intermediates key intermediates. So you can actually have A going to a completely different pathway in some circumstances. In the image that's drawn here, it's A goes to B, goes to C, goes to D. But if there were other arrows extending from B or C, those would be called key intermediates. So think of metabolic pathways like roadmaps. Roadmaps, there's multiple ways to get from one place to another place. And similarly, the roadmap of a metabolism roadmap, you can see that there may be multiple ways to get from one substance to another substance. So oftentimes what will happen is that there'll be a specific pathway that the body will generally go through, but depending on different hormones in the body, it might actually cause a reaction to go in a different direction altogether. So just think of it like roadmaps, multiple ways to get to specific end products. That being said, there are certain end products that you have to go through a certain along a certain road for, or a certain pathway for, but some have multiple paths. The way that we regulate metabolic pathways is, number one, by controlling enzyme concentrations. Quite logically, less enzyme that's present, less reaction that's going to happen. More enzyme that's present, more reaction that's going to happen. Very, very common sense regulatory mechanism for metabolic pathways. You can also have a metabolic pathway that is regulated by enzyme modulation. This is very common. And modulation of an enzyme it's a substance that will alter the activity of the particular enzyme, and in most cases it's a protein. Hormones are a very good example of enzyme modulators. So hormones, if they are released, can allow for a specific reaction to occur to a greater extent, or it could also cause a reaction to occur less than it normally would. Many metabolic pathways, while you might have a change in enzyme concentration or enzyme modulation, many metabolic pathways actually have a built-in mechanism for regulating its pathway. And this is a very common theme within human physiology. It's called feedback inhibition as a form of modulation. So if we look at this made-up metabolic pathway, you start off with the reactant A, and the end product is this substance Z. So the way that feedback inhibition works is that you go through the steps as you normally would. A gets converted to B by enzyme 1, B gets converted to C by enzyme 2, and C gets converted to enzyme Z, or the product Z, by enzyme 3. As you make more of this product Z, that itself acts as an internal inhibitor of enzyme 1. So the more Z you make, the less enzyme 1 will work. And if enzyme 1 can't work, you can't go through the steps. You can't make intermediate B, therefore you can't make intermediate C, and therefore you can't make Z. So this this is called feedback inhibition. So as you get more Z, you reduce the amount of Z that's actually being made. And then in the body, as the level of Z goes down, this feedback inhibition is removed and the reaction will continue. Eventually you reach a point where there's this constant level of Z being produced and the levels will stabilize in the blood. This is the main way that hormones are released in the body. It's feedback inhibition is a very important process, both for metabolic modulation of enzymes as well as for regulation of many physiologic processes. So this is a very important concept. Metabolic pathways are also regulated by reversible reactions. So cells can actually use the opposite reaction to regulate the direction of metabolism. And so there's 3 possible ways that we can do this. This first reaction, the first way that a reversible reaction can occur is it's the reaction between carbon dioxide and water. And carbon dioxide and water, this is a very important chemical reaction in biological systems. And this carbon dioxide and water reacting together will produce a bicarbonate ion and a hydrogen ion. You're going to see this probably a few dozen times within this course. But the important thing is that this reaction, the enzyme that catalyzes the forward reaction is the exact same as the enzyme that catalyzes the reverse reaction. And in this instance, if a reversible reaction is catalyzed by the exact same enzyme, the rule is that the system reaches a state of equilibrium very quickly and will exist in a state of equilibrium in the body. So there's going to be a certain level of carbon dioxide in the body. There's going to be a certain level of bicarbonate in the body. In this case, it only goes to this subproduct carbonic acid. But either way, the reactants and the products are in a state of equilibrium. The second method that can be used to actually catalyze reversible reactions is that there's a different enzyme. One enzyme actually causes the forward reaction to happen, and a different enzyme causes the reverse reaction to happen. These types of systems are regulated quite a bit more closely, and it's really dependent on whether these enzymes are active or inactive, or whether there's a lot of this enzyme or a little bit of this enzyme. And so the example here is the conversion of glucose and phosphate to this product called glucose 6-phosphate. This is the first step in glycolysis, which is the first step of the breakdown of glucose. In some instances, if you have a situation where the enzyme that is required for the reversible reaction doesn't exist or isn't present, this particular reaction would only occur in one direction and would be considered irreversible. So in the case of glucose plus phosphate, making glucose 6-phosphate, if you only have one of the enzymes there, you will only get one direction of the reaction. And so that reaction would be considered irreversible. So in different parts of the body, you may have only one enzyme present. And so the reversible reaction in that part of the body would be impossible. We also regulate metabolic pathways by compartmentalizing where enzymes are in the cell and which cells they are present in. So some of the enzymes are dissolved in the cytosol, and these enzymes would be the ones that are very important in glucose metabolism, so the breakdown of glucose and even the buildup of glucose. Some of these enzymes are located within the mitochondria. So the enzymes that are important in the citric acid cycle or the Krebs cycle would not be present in the same place where the enzymes for would not be present in the cytosol. There'll be different enzymes in different locations. So different parts of a chemical reaction can occur in different regions. Separation of these enzymes means that the reactions are also very separate. Some places in the body will have conflicting chemical reactions that are happening. One might build up glucose, one might break down glucose. And if you have them occurring in the exact same area, the net product is nothing. So this compartmentalization is extremely important. Fifth way that these metabolic pathways are regulated is the ratio of ATP to ADP. If there's a low concentration of ATP to high concentration of ADP, what that signals is that more ATP needs to be created. It means that the energy use within the body is very high, and you're using a lot of the ATP, so therefore it's low. And when you use that ATP, it's getting converted to ADP. So a low ATP to ADP ratio signals for ATP production to be upregulated. A high ATP to ADP ratio signifies that energy state in the body is very high and that you don't need to produce as much ATP. So this will signal for less ATP production or less breakdown of glucose. So you can imagine that the first one, where you need more ATP production, that's going to be in a state where you're going to start breaking down biomolecules to produce ATP. And in a state where there's high ATP, it means that you're in a high energy state and you can store some of this energy. So this would be in situations where you will have energy storage. And so we're going to talk about ATP a little bit now. And ATP transfers energy. It's a nucleotide. It consists of 3 phosphates, so adenosine triphosphate. And this energy is stored in the high energy phosphate to phosphate bonds. And that little tilde symbol between the 2 phosphates, that means that it's a high energy bond. And the P sub I just stands for an inorganic phosphate. So ADP plus an inorganic phosphate plus some input of energy from somewhere, maybe it's those high energy electrons, that's going to yield an ATP molecule. And the reverse reaction is going to be that phosphate to phosphate bond gets broken. There's a lot of energy stored in that bond, and when it gets broken, it produces an ADP molecule plus an inorganic phosphate, and a whole bunch of energy is released. ATP has a number of functions. It helps synthesize cellular components like proteins. for enzyme use, proteins for ion transport use, hormones. It helps synthesize fatty acids, cholesterol. It energizes muscle contraction. It energizes all the processes that utilize active transport of any kind, whether it's primary, secondary, or we haven't talked about this one yet, or tertiary active transport. It energizes all of those reactions. It energizes the secretion of hormones. It energizes nerve conduction. It energizes a lot of functions within the body. When we talk about metabolic pathways, we're going to separate metabolic pathways as being aerobic or anaerobic. Aerobic pathways require the use of oxygen, and anaerobic pathways do not require the use of oxygen. And so when we talk about anaerobic and aerobic, we're going to use these terms when we talk about the breakdown of glucose. And glucose can have a number of fates within the body. So glucose, the first fate of glucose is that it gets broken down, and we're only talking about catabolism of glucose here, so breakdown of glucose. When glucose gets broken down, the first step is that the glucose is broken into this molecule called pyruvate. And this pyruvate molecule has a few places that it can go. In the human body, it can be, if there's oxygen present, it will be completely combusted or broken down into carbon dioxide and water. That's only if there's oxygen present. If there isn't oxygen present, this pyruvate gets converted to a lactate molecule or lactic acid. And so this is the exact same stuff that is produced when you're lifting heavy weights and your muscles start burning while you're lifting the weights. That's lactic acid that's being produced in those circumstances. The third fate of pyruvate is ethanol or ethyl alcohol, or the same alcohol that is used in industry. It's the same alcohol that's used in alcoholic beverages. That process is called fermentation. This does not occur by humans. This occurs in bacteria, and the bacteria will convert the pyruvate into ethanol in the process of fermentation. So fermentation is not in humans. Only the complete oxidation is, and the anaerobic transformation of pyruvate into lactate are the processes by which glucose gets broken down in humans. And we're going to talk about all of those processes. So glucose catabolism in cellular respiration, it produces ATP from glucose, and there's really two pathways that we're going to discuss. We're going to talk about glycolysis, and that term just stands for breakdown of glucose, glycolysis. And then there is the citric acid cycle, and I probably meant to put the Krebs cycle in here, but yeah, citric acid cycle. It's also called the Krebs cycle, named after a scientist Krebs. It's also named the tricarboxylic acid cycle. You can call it whatever you want. Know all three names, but as long as you refer to it as one of those valid names, I'm fine with it. And so the glucose catabolism, we're going to start off with glycolysis, and glycolysis in general will produce very small amounts of ATP. It doesn't produce a lot of energy. The citric acid cycle also doesn't produce a lot of energy. The majority of the energy, by going through glycolysis and the citric acid cycle, the majority of the energy ends up getting released and trapped in the form of those high energy electron molecules. And it isn't until these high energy electron molecules transfer their energy that the majority of ATP is made. So very small amounts of ATP are made in glycolysis in the citric acid cycle. the majority of that ATP is made from the high energy electron molecules that are produced through those processes. So if you just went through glycolysis in the citric acid cycle, you wouldn't make enough ATP to sustain life for very long. And so here's a general overview of the process of glucose breakdown and the overall chemical reaction. So the overall chemical reaction is glucose plus oxygen, and this is the aerobic form, plus a whole bunch of ADP and inorganic phosphate, because that's what we're going to use to make ATP. That glucose and oxygen, the full breakdown of it makes carbon dioxide and water and a bunch of ATP. And if we look at it from the chemical standpoint, you get C6H12O6, or glucose, plus 6 oxygen molecules yields 6 carbon dioxide molecules and 6 water molecules. And in the process, there are between 30 and 32 ATP that are made. Different textbooks are going to tell you different things. Some are going to tell you 38, some are going to tell you 36, some are going to tell you 34. For these purposes, know between 30 and 32. So if we look at the process all the way through, note that glucose isn't the only thing that goes through these reactions of glucose to pyruvate to the end product of carbon dioxide and water. There are, at various locations within the reactions of glucose breakdown, you can have amino acids enter the cycle. You can have, so the amino acids enter the cycle and get converted to one of the intermediate steps. Amino acids can get converted to pyruvate and then go through the rest of cell respiration. Fatty acids, also from lipid breakdown, can get broken down and converted to products that can enter the citric acid cycle. Amino acids can enter the citric acid cycle. So there are many places throughout the process of glucose breakdown that other molecules that aren't glucose can enter and go through the process. So we'll end our discussion talking about glycolysis. So glycolysis meaning splitting the glucose. This particular series of reactions occurs in the cytosol of the cell, and it occurs in 3 stages. The first stage is that you trap the glucose in the cell. So once the cell has glucose, it wants to hold on to that glucose. The second stage is that you need to break down that glucose into 2 pieces, and those 2 pieces are, spoiler alert, pyruvate. And the third stage is actually making the pyruvate. So you break down the glucose, and then you have to go through a series of reactions to actually make the pyruvate. So glycolysis is actually an anaerobic process. At no place within these reactions do you actually need to use oxygen whatsoever. I want you to know all the steps of glycolysis. I'd like you to know the enzyme that catalyzes each step. And then I want you to know the specific net products of glycolysis. You don't need to know the molecular structures. This was not a fun exercise when I was in biochemistry as an undergrad, having to memorize every single one of these and being able to recognize them. I don't care if you know the chemical structures. I want you to know each step, each intermediate, and each enzyme, as well as the net products of glycolysis. That's what you need to know from this. So we're going to go through glycolysis in steps. So stage 1 is trapping the glucose in the cell. Once the cell gets the glucose, it doesn't want to give it up. And so the way that the cell holds on to the glucose, and this is a way that cells will hold on to a lot of molecules, is that they're going to add a phosphate group to it. Once a phosphate group is added, it generally will stay in the cell. And so that's how we trap the glucose in the cell. So this stage 1, there's actually 2 steps where you're adding a phosphate to the glucose. And the question is, where does this phosphate come from? And it actually comes from ATP. So you're actually needing to use 2 ATP in order to keep the glucose in the cell. And you might be thinking this actually defeats the purpose of breaking down glucose to make energy. You're actually having to use energy to start the process off. But it's just the beginning. So the first step is this glucose ends up getting phosphorylated by the breakdown of one ATP molecule. And so that phosphate from ATP ends up getting attached to the glucose molecule. And it's attached to the 6th carbon. And so that's why it's called glucose 6-phosphate. That phosphate is on the 6th carbon, if you know how to count carbons in carbohydrates or organic molecules. If you don't, that's okay as well. And the enzyme that catalyzes this reaction is hexokinase. Actually makes sense. Glucose is a hexose sugar, so hexo. And kinase, if you remember, I mentioned that anytime you see the word kinase, there's a phosphate added. And so where's the phosphate added? It's added to the 6th carbon of glucose. So you go from glucose to glucose 6-phosphate. That's the first step. Second step is you need to actually rearrange this glucose molecule. And you rearrange this glucose molecule into a fructose molecule. And that is done, and it gets converted from glucose 6-phosphate to fructose 6-phosphate. The phosphate stays on the exact same carbon of fructose, but look at the enzyme. It's phosphoglucose isomerase, and that's exactly what's happening. The phosphorylated glucose is being rearranged. You're isomerizing this glucose 6-phosphate. So the enzyme names most of the time make sense. So glucose 6-phosphate to fructose 6-phosphate. The end product for stage 1 of glycolysis is another phosphorylation step. You take fructose 6-phosphate and you use another ATP molecule, and that ATP gets converted to ADP. And that phosphate that's released is actually attached to the 1st carbon position of fructose. So you end up with a molecule of fructose 1,6-bisphosphate. And if you look at the chemical structure, you can see that there's still that phosphate on the 6th carbon. And now there's a phosphate group on the 1st carbon. And the enzyme that actually catalyzes that reaction is something called phosphofructokinase. Exactly what its name implies. The phosphorylated fructose has a phosphate group added. So it's a kinase reaction. That's the end product of stage 1 of glycolysis. Stage 2 of glycolysis is really trying to break down the glucose into 2 pieces. And that is done by an enzyme called aldolase and triose phosphate isomerase. So this fructose 1,6-bisphosphate gets converted into 2 different molecules. There's one molecule called dihydroxyacetone phosphate, or DHAP, and there's another molecule called glyceraldehyde 3-phosphate. And these 2 molecules each have 3 carbons to them, and so does glyceraldehyde 3-phosphate. And the interesting thing is that only glyceraldehyde 3-phosphate can continue on in glycolysis. So this dihydroxyacetone phosphate actually has to be rearranged to a glyceraldehyde 3-phosphate. And so the way that it occurs is this dihydroxyacetone phosphate is converted to glyceraldehyde 3-phosphate by this enzyme called triose phosphate isomerase. If you break it down, tri means 3. Ose is always for, if you remember, glucose, sucrose, ribose, any of the sugars end in ose. So this is a 3-carbon sugar, a triose. This is a triose. It has 3 carbons. Triose phosphate. Well, this triose has a phosphate to it, and you're rearranging it, so you're isomerizing it. So that's why it's called triose phosphate isomerase. You're taking DHAP, or dihydroxyacetone phosphate, and you're reconverting it into glyceraldehyde 3-phosphate. And so the end result of this process are 2 molecules of glyceraldehyde 3-phosphate that can then go through the rest of glycolysis. And stage 3 is just now making pyruvate. And so each molecule of glyceraldehyde 3-phosphate has to go through a series of reactions in order to get to pyruvate. And each of those reactions occurs twice because there are 2 molecules of glyceraldehyde 3-phosphate. That's why there's that 2 times here. So if we follow the process of glyceraldehyde 3-phosphate going through this, you are having the process by which there's glyceraldehyde 3-phosphate getting converted to 1,3-bisphosphoglycerate. And so there's a phosphate added. This is one of those reactions where you don't end up with a kinase enzyme. It's one of those exceptions. But there's also an NADH molecule that's made in the process. So NAD+, you have a hydrogen added. That's where that dehydrogenase comes from. So you're taking a hydrogen away from glyceraldehyde 3-phosphate to make an NADH molecule. This is good. This is a high energy currency for the cell. High energy electrons are transferred, are stored in this molecule. So you go from 1,3-bisphosphoglycerate, and then you end up with a reaction that removes a phosphate. And so you go from 1,3-bisphosphoglycerate to 3-phosphoglycerate. And in the process, that removal of a phosphate, you actually make an ATP molecule. Make an ATP molecule. So there's an ATP molecule that you can give back to the cell. That enzyme is phosphoglycerate kinase, and it's named after the reverse reaction, actually. But phosphoglycerate kinase is the enzyme that catalyzes 1,3-bisphosphoglycerate to 3-phosphoglycerate. And then you rearrange the phosphate group, and it goes from 3-phosphoglycerate to 2-phosphoglycerate, indicating that the phosphate group is now attached to the second carbon. It goes from the third carbon in 3-phosphoglycerate to the second carbon, 3-phosphoglycerate, to the second carbon, 2-phosphoglycerate. And the next reaction removes a water molecule. So there's a water molecule that we know is a product. This particular enzyme is called enolase. And the 2-phosphoglycerate in the presence of this enolase makes phosphoenol pyruvate. And then the very last reaction is the removal of the last phosphate, and you make another ATP molecule. And that's catalyzed by pyruvate kinase. So you remove that ATP, and you go from phosphoenol pyruvate to pyruvate. These reactions occur twice. So for every single glyceraldehyde 3-phosphate, you're going to get 1 NADH, you're going to get 2 ATP, you're going to get 1 water, and you're going to get 1 pyruvate. And so each glucose molecule, be very careful about what we're talking about here. For every glucose molecule, you actually have 2 glyceraldehyde 3-phosphate molecules going through. So all of these numbers would be doubled for each glucose. So let's balance the energy products of the energy and the products of glycolysis. So at the end of glycolysis, each glucose molecule utilizes, well, the first stage, remember, we use 2 ATP. We use 2 ATP. But then the glyceraldehyde 3-phosphate to pyruvate, well, we made 2 pyruvate. We made 4 ATP altogether, 2 for each glyceraldehyde 3-phosphate. We made 2 NADH molecules, and we made 2 water molecules. So the balanced reaction for glycolysis is 1 glucose yields 2 pyruvate plus 2 NADH, and then 2 waters, and 2 net ATP because we used 2, but then we made 4. So we actually get 2 ATP that are made already. When we look at the reactions of glycolysis, most of them are reversible. Most can go in either direction, but there are a few that are relatively or mostly irreversible. And these irreversible reactions generally will have a regulatory function within this process of reactions. And there are 3 mostly irreversible reactions within glycolysis. There's the hexokinase step, so that's the very first step of glycolysis, whereby you took glucose and you added that phosphate group to the 6th carbon and made glucose 6-phosphate. There's the phosphofructokinase step that took fructose 6-phosphate and made fructose 1,6-bisphosphate. And then there's the pyruvate kinase step that took phosphoenol pyruvate and made pyruvate. So if we look, the most important regulatory step is actually the phosphofructokinase step. Although all of these are relatively irreversible, phosphofructokinase is the most important regulatory step, and there's a reason for that. So the reaction between fructose 6-phosphate and fructose 1,6-bisphosphate, that's the one that requires an ATP molecule, and it adds another phosphate group to the first carbon. The activity of this phosphofructokinase is very dependent on the level of ATP. Very high ATP concentration will signify that this enzyme does not need to work as much. So the cell doesn't need to make energy, and you can actually store the glucose. High ATP concentration in the body signifies that you're in an energy surplus state. Low ATP concentration, on the other hand, gives this phosphofructokinase step high activity. The cell needs to make more energy. More glucose needs to go through this process. And this enzyme is going to function quite a bit with higher activity. But the important part of this phosphofructokinase step is that once you make fructose 1,6-bisphosphate, this product can't go through any other metabolic pathway to break down and make energy. Once you make this product of fructose 1,6-bisphosphate, you are committed to going through the rest of glycolysis. You cannot go in reverse. Hexokinase, on the other hand, is a mostly irreversible reaction, but this particular reaction is not as important as the phosphofructokinase step in regulating glycolysis, because you can go from fructose or glucose to glucose 6-phosphate using hexokinase, but you can actually go in the reverse direction using a completely different enzyme. There's another enzyme that helps you go in the reverse direction. With phosphofructokinase, there is no reversible enzyme. And so phosphofructokinase is the most important regulatory step, because once you go through the phosphofructokinase step, you are obligated to finish glycolysis. With hexokinase, that glucose 6-phosphate, it can be used in another metabolic pathway. It's actually used in the metabolic pathway that stores glucose as glycogen. And so in general, when we talk about metabolic pathways and we talk about the steps of metabolic pathways, the enzyme that catalyzes the step that is committed, or that, for example, that phosphofructokinase step, you are now committed to going through the rest of glycolysis. The committed step is the most important control step. And so that's a general rule of thumb for almost all metabolism. We'll end there today, and we'll talk about the citric acid cycle, or the tricarboxylic acid cycle, or the Krebs cycle next day. We'll also discuss the electron transport system, and we're going to talk a little bit about metabolic rate and control of metabolic rate. I'll see you next time.